Up until this point, you've learned a lot about the importance of DNA and what it does, and how it can help with genes a little bit. We don't really know what it is just yet, and how it leads to traits on our body. So this first lesson here is about the structure of DNA, so we can get to know a little bit more about what constitutes DNA and what it looks like. Well, DNA happens to be a double helix, which means you've kind of got this twisting band with staircases across the middle. And these twisting band, this red part here, is kind of the backbone that holds the rungs of the ladder together. And the two things that make up the backbone are deoxyribose, which is where DNA gets its name from, deoxyribonucleic acid, and phosphate. And they alternate going up and down these chains: deoxyribose, phosphate, deoxyribose, phosphate, deoxyribose, phosphate, etc. Notice deoxyribose is a sugar. If you remember, these empty spots here are typically C's, so we just have carbon, oxygen, and hydrogen because it's a sugar. And phosphate is just a small molecule, PO4, made of phosphorus and oxygen. As you look at the backbone here once again, notice here's the deoxyribose, the phosphate, the deoxyribose, the phosphate. They call it a sugar phosphate backbone here. Part of the reason they do that is because in RNA you have ribose and phosphate alternating, which is also a sugar and phosphate backbone. And in the middle here are our nitrogen bases. And so one sequence here of a nitrogen base, a deoxyribose, and phosphate is called a nucleotide. And a nucleotide is really the monomer that makes up the polymer that we call a nucleic acid. And DNA is deoxyribonucleic acid. As you look here, you'll see there's 4 different nitrogen bases that make up DNA. There's adenine, thymine, guanine, and cytosine. What you may notice as you look at these is that 2 of them kind of look alike, these 2, because they have double rings, and 2 of these kind of look alike because they have single rings. Now those are carbons that are in there, and notice once again all that nitrogen. That's why they're called nitrogen bases. Well, these double ring ones are referred to as purines, and our single ring ones are referred to as pyrimidines. And that's a way these nitrogen bases are sometimes classified, because in order for this staircase to work, you can't have one set of rungs with 2 rings and then the next set of rungs with 4 rings, or else your staircase is going to be a bit wonky. So what happens is cytosine and guanine always pair up. That way you always have the 1 ring from cytosine plus the 2 rings from guanine. And adenine and thymine always pair up, which is why you have the 2 rings from adenine and the 1 rings from thymine. And so what that does is it ensures as you go down the DNA ladder, you've always got 2 rungs on one side and 1 rung on the other. Now they could be matching up like this, but this allows that staircase to stay straight. And one of the ways they found this out is once they were able to break down DNA, they found there were typically equal amounts of guanine and cytosine and equal amounts of adenine and thymine, and so they knew that those are the ones that paired up together. And you'll often see these abbreviated as A, T, G, and C. So as we put everything all together, we'll refresh. This is called a nucleotide, which is our monomer that makes up the nucleic acid. You've got your backbone of deoxyribose, phosphate, deoxyribose, phosphate, etc. You've got your paired nitrogen bases here. C always goes with G, G always goes with C, A always goes with T, T always goes with A. Now what holds them together here, right between the phosphate and the deoxyribose, are covalent bonds, which means that they are sharing electrons. You might remember that from physical science. Sharing valence electrons. That's why it's covalent. And notice here between your different nitrogen bases you have hydrogen bonds. The C and the G have 3 hydrogen bonds holding them together, while adenine and thymine only have 2 hydrogen bonds holding them together. But that's still strong enough to keep this staircase together. But what it also means is that this can get unzipped relatively easy, which is going to be a key important factor when we try to replicate DNA. It's really important that these covalent bonds between deoxyribose, and phosphate, and deoxyribose and the nitrogen bases, it's real important that these covalent bonds don't break, but we do want this to unzip right down the middle when we try to replicate DNA later. So there's the structure of DNA. You can see the spiral staircase here because it is a double helix. There are 2 sets of this backbone, one on each side, and nucleotide on each side that is paired together. So covalent bonds hold this sturdy part together, and hydrogen bonds down the middle so that we can unzip that down the road when we need to replicate DNA.